Hello everyone, I am Neenu MK, Assistant Professor, Department of Pharmacognosy, Yenepoya Pharmacy College and Research Center. Today for this session, my topic is isolation and preservation method for pure culture. The table of contents will be coming, what is pure culture, what are the isolation methods and what are the preservation methods for pure culture. Specific learning objectives. By the end of the session, I think you will be able to know about what is a pure culture, different methods for isolation of a pure culture, then different methods for the preservation of the isolated pure culture. Then before going to the isolation and preservation, first we need to know about what is a culture, what is the difference between mixed culture and the pure culture. Culture means it is an act of cultivating a microorganism or microorganism that which we are cultivating. That is culturing, and the microorganism also we can call it as a culture. Then difference between mixed culture and pure culture. Mixed culture means from the name itself more than two or more than one microorganism will be there. Pure culture means it will be having only a single species of organism. Then microorganism, it will not, in nature it will not be in the pure form, it will be always in the mixed form only, or the mixed culture will be there. So from mixed culture we will be taking out the pure culture, or from that we will be subculturing and or isolating out the pure culture. Then coming to the main isolation techniques, which and all are the isolation techniques for taking out a pure culture. Strict plate method, pour plate method, spread plate method, micromanipulator method, roll tube method. In pour plate method also two types are there, loop dilution and serial dilution techniques. Strict plate method, it is a commonly used method, and how we will be performing means, as in the picture, first from one end to the another end we will be streaking out the mixed culture. First small amount mixed culture will be taking by using an inoculation loop and streaking on the solidified agar medium. Thus successive streaking will leads to the thin out of the inoculum. So that dilution will be forming and we will get a thinned out, or the discrete colonies we will get on the successive streaking. Because of this dilution gradient, confluent growth will not be there, only few cells we will be getting. Thus, we will be adding to the fresh medium and taking out the pure microorganism. Then in streak plate method only, different types of streaking will be there. That is quadrant streaking, radiant streaking, T-streaking, continuous streaking. First one is quadrant streaking. What is a quadrant streaking? As the name, quadrant means four areas will be there. So plate will be dividing into quadrants or four regions, and sample will be successively streaking out from one quadrant to the another one. So first the stock culture or the original microorganism which we will be taking and diluting on the successive streaking. From the first area then to the second one. In first area, if we are streaking means, after coming to the end of the first area, we will be taking out the inoculation loop and making it red hot. Then after cooling from that first end to the next second part, we will be again streaking. Then from the second, after making it red hot, then after cooling, from the second to the third end, then fourth end, after that it should not touch the first end. This is about the quadrant streaking. Then next one is radiant streaking. Radiant streaking means first in Petri plate or the solidified agar Petri plate we will be taking, in that by using inoculation loop from one area, that is area 1. From there this inoculated inoculation loop, flamed inoculation loop will be taking and keeping or doing 7 to 8 straight lines streaking we will be performing from the area 1 to the opposite end. In the picture, area 1 is there, from that to the next end 7 to 8 straight lines will be streaking. Then after that again this loop will be flaming, and after cooling down then opposite or cross down again we will be streaking to the 7 to 8 straight lines, crossing down of the another straight lines, or another straight lines crossover over the 7 to 8 lines will be performing. That is about the radiant streaking. Then the third type of streaking, that is T streaking. As the name, T means we will be drawing a letter of T on the back side of the Petri plate, which is having that Petri plate should be having a solidified agar medium. In that solidified agar medium, backside of the Petri plate, we will be drawing the letter T. Then because of drawing that T, that Petri plate as medium in the Petri plate will be dividing into 3 sections. First section will be the upper portion, then down 2 sections will be there. So in the first section we will be streaking, then from that first to next down 2 sections, from that 1 section, then from that to the another section. So 3 sections we will be streaking successively. This is about the T streaking. Then continuous streaking. Continuous streaking also Petri plate we will be taking, then making it into 2 areas, that is area A and B. So in that Petri plate, by using inoculation loop, from the area A to the middle of the or the center of the Petri plate we will be streaking. Then after streaking till the center, this plate is rotating at 180 degree. Then after rotating means we will, then the inoculation loop next we need to keep at the B area. This Petri plate B area we need to keep the inoculation loop. Then from that B edge to the center of the Petri plate again we need to streak. So it will become like an continuous from A to center, then turning it in 180 degree and from B to center. This is about the continuous streaking. So in isolation, first method we have finished, that is streak plate method. In that 4 types of streak plate methods are the quadrant, then radiant, T and continuous streaking. Then coming to the next type of method for the isolation of pure culture, that is pour plate method. In pour plate method, this is for isolating, and also we can count out the viable microorganism which is present in the liquid sample. How we will be performing this one means, first we need to prepare the microbial sample. Microbial sample means first it may be in the semi-solid or solid form, and also it may be in liquid form. If it is in the semi-solid or solid form, we need to suspend it in the sterile water or in the nutrient broth, agar medium, or that medium or that broth we need to make it into a broth type, or liquid type we need to make, liquid solution we need to make. If it is already in the liquid form means we need to prepare serial dilutions. Why we were doing serial dilution means in order to decrease the microbial colony load, which means from 10 raised to minus 1 to 10 raised to minus 10, till that we need to dilute. First this microorganism will be taking and 10 raised to minus 1, that is 1 ml we need to add microorganism. Then from that first test tube to the next test tube, from that 1 ml we were adding to the successive test tubes, and it will be till 10 raised to minus 10 test tube we need to make. This is the sample preparation. After sample preparation, how we can prepare or how we can make this pour plate isolation technique. 2 methods are there. One is, first we can add this, which one, this prepared sample or this prepared microorganism directly we can add onto the Petri plate. Then nutrient agar we can pour onto the added microorganism containing Petri plate, then swirl to mix, then we can incubate. That is one method. Or else we can do both mixing, we can perform it before only, like microorganism and nutrient agar we can mix before only, then that mixed one we can pour to the Petri plate and then keep it for incubation. After incubation, colonies will start appearing on the surface, and also middle or beneath the medium also we can see in this method, because here we were mixing. So in this method, what is the difference means, here down or beneath the medium also we can see the growth of microorganism, but in streak plate method and all, only at the surface we can see the microorganism growth. Then next method is spread plate method. Spread plate method means here we were adding the microorganism on the, or the inoculum on the surface of solidified agar medium, but that adding directly we will not add, we will be diluting the microorganism. Diluting in pour plate method also we were diluting, but it is not like the pour plate method. Here what we are doing means we were diluting by using sterile liquids such as water or physiological salines we will be using. But in pour plate method, what we are using? Melted agar medium or broth we were using, agar we were using. Here liquids we were using, and like water and physiological saline we will be using, and spreading by using a bent glass rod, one bent glass rod as in the figure here, as like this. We will be using an bent glass rod, and this bent glass rod will be helping to spread out the inoculum which we kept on the Petri plate. That is about spread plate method. Then the next one is micromanipulator method. Micromanipulator is an instrument, or it is a device which is used to physically interact with the sample under a microscope. So this instrument is in conjugation with which one? With which? The microorganism. Why we microscope? Why we are using microscope means to pick up the single cell of microorganism that we are taking from the hanging drop preparation which we kept on the slide, cover slip which we kept. From that we were taking the microorganism single cell by using the microscope. So micromanipulator, in that micrometer adjustment is also performing by means of the micropipette. This micropipette which we can move right, left or forward, backward and up and down by the help of micrometer adjustment. See, this is the instrument this is the device, micromanipulator device. Here micropipettes are there. This pipette will be helping to take out the single cell and transferring to the another glass slide which is having medium. So which cell we need to isolate, or the pure cell we will be identifying by the help of microscope. Then after find out the single pure cell, that cell we will be taking with the help of micropipette. Then from there to the another by the section, this cell is drawn by using micropipette and transferring to a large drop of sterile medium on the another slide, sterile cover slip. So this is about the micromanipulator method. Then the last method in isolation of pure culture is roll tube method. This is another variation or the modification of streak plate method. Here also we were streaking, but what is the difference means here anaerobic bacteria we were isolating out. We were giving an anaerobic condition. Here stoppered anaerobic culture tube will be using, and that culture tube inner wall will be coated with pre-reduced media, and it is filled with oxygen-free carbon dioxide by the help of gas cannula. Then how we will be streaking or how we will be performing the inoculation means, inoculum will be taking in an inoculation loop and keeping held against the agar surface, and we will be owning the motor. With the help of the motor, this tube will be rotating. Based on that, this inoculum will also spread over the tube. From bottom to the upside it will spread, and this inoculum also become thinned while coming upside. And then this tube is restoppered and keeping for incubation. Then microorganism will grow and we can isolate out the single pure cells. This is about the different methods for the isolation of pure culture. For 5 methods we have discussed. Then coming to the next topic, that is this prepared or this isolated pure culture will be there, that we need to preserve. How we can preserve these prepared this isolated pure culture? Preservation, why we are performing means, in order to maintain this pure culture for an extended period in the viable condition only. There should not happen any genetic changes, should not happen over there. For that we were we were preserving this isolated colonies or isolated culture we were doing. What is the objective of preservation? First, as we told, it is to maintain the isolated pure culture for an extended period in a viable condition. Then contamination should be avoiding. Then genetic mutation should not be there. If we are preserving means it will restrict the genetic mutation. So coming to the different preservation methods, what and all are the different methods? First one is periodic transfer to the fresh medium, then storage at low temperature, then storage in sterile soil, preservation by overlying culture with mineral oil, lyophilization or freeze drying. This is the 5 techniques for the preservation of isolated pure culture. In that, first one is periodic transfer to the fresh medium, which means from the old stock culture, which means already isolated culture will be there. From that, we were taking the microorganism and transferring to the fresh nutrient medium. It is subculturing process. Here it is happening as a subculturing process. Then next one is storage at low temperature. Low temperature storage means refrigeration will be there. As we all know, refrigeration means it will be keeping at the temperature of almost 0 to 4 degrees Celsius in cold room or in refrigerators. Then this is actually for short duration. Normally 2 to 3 weeks. If we are taking bacteria means it will be 2 to 3 weeks, or if fungi means 3 to 4 months it will stay. Because why this much of short duration means metabolic activities may happen. Here growth will not be stopping. Instead of that, it is decreasing the growth. Microorganism growth is decreasing, so this metabolic activities may happen, so that this is only for a short duration. Then next low temperature method is cryopreservation. Cryopreservation means by using the liquid nitrogen, freezing in liquid nitrogen at almost minus 196 degrees Celsius, or in the gaseous phase above the liquid nitrogen at minus 150 degrees Celsius. This will be keeping for long storage time. This we can keep for long storage. How we will be doing means, if we are doing like this or freezing means, we need to add dimethyl sulfoxide, that is a DMSO we needed to add. That is acting as a stabilizing agent, because when we are freezing or the crystal if it is forming means, there is a chance of cell damage for the microorganism. So in order to prevent that, we were adding DMSO and all. Then almost all species, if we are doing this type of preservation means, it will be keep it for 10 to 30 years we can keep this one in case of cryopreservation. Then the next method is, preservation method is storage in sterile soil. We can mix the we can mix the inoculum with the sterile soil and dry, then keep in refrigerator. That is one method. Or else simply soil, with the soil we can mix the organism and keeping incubating that soil with the organism means that organism will start growing. That condition we will grow in like mycelium and myceliums and propagative unit of second generation and all will be preserved by this method. So soil preservation mainly will be using for fungi. Then by this method actinomycetes are maintained for almost 4 to 5 years. Then the next method is preservation by overlaying culture with mineral oil. Here what we are doing means this sterile liquid paraffin will be there. Above the culture we will be pouring and keeping it in the upright position at room temperature only we will be keeping. So first we will be adding the liquid paraffin over this one. So here we were finding out an anaerobic condition we will be creating. So dehydration will not happen over here. This medium or inside or below the liquid paraffin, medium will be there, right? That medium will not dehydrate when we are adding the paraffin, liquid paraffin if we are adding. Then the next method is lyophilization or freeze drying. Freeze drying means it is a stabilizing process. Here first we were the substance first we are freezing, that freezing will be happening, then the quantity will be reducing. So first sublimation will be happening, then desorption will be happening. So here freezing and dehydration is combining. So first initially frozen and then drying by lowering the atmospheric pressure with a vacuum apparatus. This is a better method when comparing to the freeze drying, because here we were because of freezing that risk will not be there in this method. So these are the preservation methods. We discussed about the isolation methods and preservation methods. So isolation methods will be coming: streaking, pouring, spreading, micro manipulation method, and the roll tube method will be the isolation techniques for pure culture. Why we are performing isolation means in order to ensure the accurate microbial identification and study. Then after isolation, we will be preserving this. Preservation is mainly for maintaining the viability and preventing the contamination or genetic changes in culture. So proper preservation if we are doing means it will extend the usability of the culture for research and industrial applications. Advances in this both preservation and isolation technique can enhance the reproducibility and short-term or long-term storage capabilities also. So this is about the isolation and preservation techniques. We have discussed what is culture and what is the difference between mixed and pure culture, and what are the isolation techniques and preservation. These are my references. Thank you.